Swayam Prabha. Digital India. Educated India.
Okay, while on this, one point I did not specifically talk about while we were talking about specification. Remember, we talked about autonomous specification leading to mosaic development, then conditional specification, regulative development, then we also looked at syncytial specification. So, okay, so these are three different modes. The point that I missed is are these modes mutually exclusive? Like for example, if one organism follows autonomous specification, is it all through autonomous, or if something follows conditional, is it all through conditional? The answer is no, it is a mixture. So, most of the, like for example, in vertebrate development it is both. So, initially it is conditional, later in specific lineages you have autonomous specification. So, we were at this step, right. So, here we stopped, like how the, no, we have not gone there. So, we stopped here. So, how the localization of gherkin mRNA in this posterior side of the nucleus, posterior being closer to the follicle cells, that is the reference here. And this gherkin protein binding to this torpedo receptor on the follicle cells ends up signaling that for terminal follicle cells to acquire a differentiated fate. So, they become not undifferentiated follicle cells, now they are terminal follicle cells, and then they signal back, and as a result of that signaling you have this micropolarized microtubule formation resulting in bicoid on the anterior and oscar at the posterior. And one of the key components of this signaling from follicle cells to the oocyte is this localization of PAR1 protein. So, once this anterior posterior polarity is established, so let us not focus too much on the molecules and forget about the developmental stage. Okay. So, here we are in oocyte right now, no fertilization has happened. So, in the oocyte itself the anterior posterior is polarized. Now, along these microtubules the nucleus, oocyte nucleus migrates anterior on the dorsal side. Okay. So, this way, and the gherkin goes along with the nucleus and finally gets localized to this corner, okay, anterior dorsal side. Okay. So, this is showing the bicoid by in situ hybridization, bicoid mRNA. So, the nanos mRNA gets localized through multiple mechanisms, the localized, like the one that gets localized gets protected from degradation, the one that is not localized gets degraded, and only the localized one gets translated, all that. So, that we will see later when we are going to look at the anterior posterior axis formation. So, for now let us focus on the gherkin some more. So, this is in the in the oocyte, this is the whole egg chamber and this is the oocyte, these are nerve cells, and this is where you have the gherkin mRNA, and this another look at the same. And if you make a cross section, the follicle cells, the dorsal, and then here you have this. So, this is protein there. So, what do you think is this red color? Based on whatever we have discussed earlier where we have seen red color, good progress, cytoskeletal, one of the cytoskeletal, actin. So, actin is what is there in the inner periphery of the cell membrane. So, it kind of makes the boundary, so therefore, it is actin. Alright, so here is an experiment. So, we could have ignored this and moved on, and this is just to get an idea of how did people understand this torpedo gherkin signaling. This is the manner in which it happens, like it is on follicle cells and they signal back and so on. So, this scheme kind of tells you where do you think torpedo should be produced? From the oocyte nucleus, or should it be produced in the follicle cells? So, which one should contain the genetic information for torpedo protein making? Which one, follicle nuclei or the oocyte nucleus? Follicle one. So, which means if I did a genetic manipulation such that this oocyte genome has a torpedo mutation, but it is in a mother, like the somatic cells coming from the mother, that is not torpedo mutant, then it should develop normal, but the opposite will not develop normal. So, that is this experiment. So, here you have pole cells, okay, the wild type. Pole cells are the germ cell precursors, so primordial germ cells. Okay. When the syncytial embryo is going to make cells, like cellularization happens, the first cells made are these pole cells. They kind of come out of the main part of the embryo. Okay. So, the pole cells are here. So, these are the ones from which you are going to make oocytes, right. So, that is why we are going to focus on that. So, we are going to take the pole cells of the wild type mother, okay, and then transplant to an embryo that is deficient in torpedo gene. So, here this is not going to produce torpedo, but the germ cells that are going to form from that are going to be normal and swapping in the opposite direction as well. So, torpedo mutant germ cells you are putting in the wild type, and now you allow this embryo to develop into adult fly. Okay. So, here you are going to have this. So, this is going to make the follicle cells, germarium, etc., based on its genotype. Okay. Similarly, this one. So, this is going to be making follicle cells that are not going to have torpedo protein, while this one is going to make pole cells or the oocyte, it is going to make oocyte. The relevant thing here is oocyte, which will not have torpedo protein producing ability, but the follicle cells, they will have the torpedo. Now, here you have torpedo deficient oocyte in wild type follicle, and that is going to make normal dorsoventral axis, okay, because torpedo is produced in the right place for the polarization to happen, and the opposite is the case with this. So, here the follicle cells are not going to produce the torpedo, although the oocyte is totally wild type, and that is not going to develop normally. Okay. So, this is the kind of transplantation experiments with the germline chimera, like you have chimera because one, like the somatic part is one genotype and the germline is another genotype. So, this kind of proved that model that we just saw, right. So, they are taking the pole cells. So, that does not affect this embryo in any way, because these have come out of the embryo. So, these are isolatable cells without affecting the embryo developing into a normal fly, just that fly will be sterile because it will have no germ cells. So, you take those cells and transplant to an embryo that is developing where the mother is torpedo mutant mother. So, here the embryo is going to develop into an adult, the somatic part of the adult, that will not have torpedo. So, if you open its ovary and ovariole and go to the germarium and egg chamber where follicle cells are developing, there will be no torpedo, because this all of them is somatic, and that somatic is torpedo mutant. But the germ cells you took from wild type and placed when it was embryo, and they are there. So, they are the ones that are migrating to the somatic gonad and establish the germline there, and they are making the stem cell division and the cystoblast becoming 15 nerve cell oocyte, all other happening from these. Okay. And now, in that egg chamber, so in that is like keep this in mind, then we go here. Okay. here, yeah. So, one more. So, these are not having torpedo, while this nucleus has the wild type copy of the torpedo, and that is of no use, it has to be produced here, and that is what is this. So, as a result, this does not have that axis formation. So, this embryo comes from a mother whose genotype is torpedo mutant, okay, and as a result whatever she is producing, she is not going to like, okay. So, we go little earlier, you take this stage or even earlier, okay. So, all of this is happening in that mother, and that mother is not going to have this with the torpedo. So, this genotype is that mother's somatic cells, but the germ cells, when this was in early stage, you actually put the wild type germ cells. See, they took advantage of the fact that these pole cells come off separate and they can be taken out, and as a result in this diagram these cells are going to be like normal, these are not torpedo mutant. So, I am particularly pointing out these are not the oocytes, but they are germ cell origin and they are all wild type, only this is not going to be, and so that is what they are illustrating by coloring the whole fly of that torpedo mutant color that we associated with. So, these kind of creating chimera, where this is in a sense is kind of mosaic, like what you are trying to do is the genome of different cell types in the same organism are not identical, okay. So, here in this you have two different genome, like one of genomes, one of them has wild type copy of torpedo while the other one does not. So, the question is where the torpedo has to be produced for this dorsoventral axis formation. Okay, this is going to be a lot more complicated if you thought that was complicated, okay, but I will take you through very slowly and it is not going to be complicated. So, here what we are actually going to do is, so remember, let us not lose the big picture. The big picture is, our goal of this series of lectures is to understand how do we make front and back and top and bottom, okay, asymmetry in an embryo which starts in a symmetrical zygote, right. So, the example we took is Drosophila, where this asymmetry is made in oocyte itself, and now we focused on in oocyte how the asymmetry happens. So far we saw the anterior posterior asymmetry in the oocyte. Now we are going to switch gears and look at top bottom, that is dorsal ventral. Okay, top bottom does not mean head and tail in us who is standing on two limbs. For us it will be front and back. Okay, so your back is top and your front is ventral. Okay, dorsal is back, ventral is belly side. Clear? So, the animal is like this normally, only we are standing straight, and head is anterior or front and tail is back. So, in that sense your head is like the front. So, keep this. So, many times developmental biology classes, they assume that everyone comes from biology background and therefore these are all elementary and they do not talk about it, textbooks do not talk about it. But if these people come and teach in India, they will realize all biologists are actually non-biology origin, transplanted ones, and therefore you have to emphasize this, okay. So, for example, in the fly, when I am saying this is front, for you this is front, okay, the analogous structure is here, right, and this is top. It is not this, it is this back, and this is not bottom, this is front of your body. Clear? You will not get confused, right? Okay, this is very important. Alright, so now we are going to look at dorsal ventral asymmetry, how that is made. So, it is also called axis, okay, anterior posterior axis we saw, and now we are going to see dorsoventral axis. And this one, it starts in the oocyte, but we are going to end up with embryo. So, it is continuously moving, like when I am starting here I am in the oocyte, but when I am going to be here it is going to be syncytial embryo. So, while this nucleus is oocyte nucleus when I am going to start, when I am going to end, this is syncytial nuclei, among many nuclei that is made, this is one of them and which is on the ventral side. Okay. So, pay attention to this label, here it is written correctly, okay. I have edited here, you can see that color difference, I could not match this color correctly. The book says oocyte there, okay, do not get distracted by that. This is a blastoderm embryo when we go to that stage. Okay, so now we will go slowly. So, remember when the nucleus I said moves to the anterior on the dorsal side and gherkin goes along with that as a halo, right. Now, this gherkin is going to do something. So, it is all that gherkin we started with that is going to be important for both the axes. So, now we are going to focus on that gherkin, what it is going to do. Yeah. So, that gherkin mRNA localized here is going to produce, like the approximate location is here, okay, the dorso anterior, and that is going to go and bind to torpedo receptor on the follicle cells here. Remember, torpedo is all over, it is just that in the previous temporal stage it is the terminal follicle torpedo that received the gherkin signal. So, now the torpedo on these dorsal follicle cells, they receive the signal, and that signal stops the production of a protein called a pipe. This is at the bottom, okay, the spelling if you want, pipe, the same as normal pipes. So, the pipe is not produced here. So, the dorsal follicles do not produce pipe as a result of gherkin signaling through torpedo receptors on the dorsal cells. While that does not happen in the posterior, because in the posterior you do not have gherkin. So, the posterior follicle cells do not have the torpedo signaling, therefore, they do not inhibit pipe synthesis. So, they are going to make pipe. Now, we are going to zoom and look at this, like this area if you zoom, where you have the oocyte cytoplasm and the perivitellin space between the two vitellin membranes and then the follicle cells. So, this is the zoomed up here. The only thing which I have not corrected is, you look at this receptor in this clearly drawn on the follicle cells, and here it is drawn on the oocyte. Okay. So, and same color, same shape, and you assume this is tall receptor drawn here, but actually and tall receptor is what we want to focus, therefore, this should have been drawn on this side as it is drawn here. So, this is correct. So, now let us see what the pipe made in the ventral follicle cells is going to do. That pipe is going to associate with a protein that is still not identified, and that is going to come out, and the way our blood clotting system gets activated in a cascade of proteolytic cleavage, it is going to have these cleavages. Okay, this is going to associate with a protease that gets activated, cleaves this gastrulation defective protein, activated protease, cleaves snake, activated snake protease, cleaves ester, activated ester protease, and that is going to cleave a SPAD cell to an activated SPAD cell, which is not a protease, that is going to bind to TOL, and the TOL is going to transduce a signal from the SPAD cell. Okay. All of this happens in the perivitellin space, in that tiny space there. By the time all of this happens, the oocyte is fertilized and it has gone to syncytial blastoderm. Okay. So, syncytial blastoderm is, do I have an image for that? If I do not, I am going to draw one. No, I don't. I will have, right? Syncytium we talked about. Yeah. This is a good one. This helps. So, syncytial blastoderm means that fertilized zygotic nucleus is going to divide synchronously 13 times and generate lot of nuclei. So, initially the nuclei are going to be everywhere and later they localize to the cortex, and that stage is what we are looking at here. Syncytial blastoderm where the nuclei have migrated to the cortex. So, the cortex is the area just below the cytoplasm. So, that means the outer area of cytoplasm. Anything in biology, the outer area, but inside some boundary is what is cortex. Okay. So, it is cortical localization of these nuclei. So, that is the stage we are when we are talking this step. So, now this is one such nucleus that is at the cortex, but on the ventral side. See, suppose if this is a cell, this area is cortex, the outer part, periphery, but inside the membrane. So, in the oocyte, let us say the dorsoventral axis is primarily the asymmetric activation of torpedo. So, the dorsal follicle cells do not make pipe while ventral follicles make pipe. So, this is how at the oocyte level dorsoventral axis is made. Now, this asymmetry, molecular asymmetry, the pipe being produced in the ventral follicle cells becomes consequential when you reach the blastoderm embryonic stage. Okay. Because this is happening in a span of about 90 minutes. So, we are not immediately considering the same. Okay. So, by the time this asymmetry has a consequence on development, this nucleus has divided multiple times, 13 cycles, generated lot of nuclei, and they arranged on the cortex, and now the ventrally located nuclei are getting influenced by the asymmetry generated during oocyte. So, the consequence of the Tal signaling is the Tal signaling leads to activation of a kinase, this pelle, which probably requires tube is what they say. We do not exactly know how that influences. That is going to phosphorylate a protein called cactus, okay, and that cactus is going to be degraded. Phosphorylated cactus is going to degrade. And I need to back up in terms of time scale and then introduce these two players here. So, dorsal is a protein produced from the maternally encoded mRNA a little later during embryonic development, okay, not in the oocyte stage, but in the zygote it gets translated into protein, and that protein is throughout the cytoplasm. Okay. Syncytial cytoplasm, all over you have the dorsal, but it is in the cytoplasm all over, and that is because this cactus protein binds to it and prevents it from migrating into nucleus. Otherwise, it has a signal that can take it to the nucleus. But in the ventral side, in the ventral cytoplasm, because of this signaling triggered by the pipe synthesis in the ventral follicle cells, the cactus is going to be degraded, and as a result free dorsal is high in the ventral cytoplasm. So, it enters into the ventral side nuclei, and that is going to trigger a chain of transcriptional events, and as a result the dorsal nuclei have a new set of genes activated or inhibited, and that is what is going to specify those nuclei to become ventral nuclei. So, it is little bit more, but I will tell in one sentence and then we will move into the detail. The point is the dorsal activation in the ventral nuclei is going to activate genes required for the formation of ectoderm structures and also inhibit genes required for the formation of other structures that will be on the dorsal side. Okay. So, it is going to do two: activation, inhibition both. Activation to make the ventral side, inactivation of the genes required to make the dorsal side. So, this is a experimental evidence, here dorsal protein mutant. If you do not have dorsal protein mutant, then you do not make this nice exoskeleton, instead it is kind of all haphazard, messed up. And this is the summary of that, but we will get into the details. So, the ventral nuclei that received the maximum of dorsal, okay, we are not talking about dorsal in general, dorsal inside the nucleus, okay, dorsal is there all over the cytoplasm, but it is a transcription factor, it has to get into the nucleus, and that happens only in the ventral side, and that is going to become the mesoderm, and then slightly lower is going to become ectoderm, and then these ectoderms, and the lowest is going to form the amnioserosa. Those what is amnioserosa, you need not worry, all that matters is ventral dorsal specification is made by the dorsal protein gradient. So, why the name dorsal for a protein that makes ventral? That is because of the mutation, okay, the phenotype of the mutant, ventralized, okay, sorry, dorsalized, therefore it is called dorsal. You do not make something like a mutant that does not make germ cells is called germ cell less. It is not that the encoded protein is going to make the germ cell, it is germ cell less. So, the names are based on the phenotypes. So, dorsal protein is required to make the ventral. So, in its absence the embryo gets dorsalized. So, therefore, it is dorsal. You look like a dorsal, so you are called dorsal. So, now you look at this wild type dorsal, plenty of dorsal in the nuclei. So, now you see this the cortical arrangement of the nuclei I was telling about, plastoderm embryo, right. So, here you have these white round structure are the nuclei all over. So, this is in the this is cortical localization. So, this is central cytoplasm. So, here you have dorsal protein, and in a mutant, one of those these things, if you mutate any of these, I do not know exactly which one, the legend in the book will tell you exactly what is the mutant, and in that mutant dorsal does not get into the nucleus, and as a result no ventral specification, the whole embryo is dorsalized. And in another mutant where you have it everywhere, then that embryo is ventralized, right, I guess so. So, I do not exactly know what, maybe Gherkin localization is affected, or they injected Gherkin mRNA to the ventral side. So, it could be any of that. So, but the legend specifically tells you what is that mutant. So, I just do not remember right now. So, now what is the consequence of this? How is it getting ventralized? So, remember the ventral furrow formation that we learnt when we were learning about morphogenesis, right. So, that is coming back here. So, this is just a box item, we will get to our main story right away, but one of the events is a particular transcription factor called twist is expressed only in those cells, that is going to rearrange, remember the beta catenin, myosin, all that, and as a result you are going to have apical constriction, and that is leading to this sort of a thing, and finally for the cells migrating inside and making a tube like structure, right. And that is explained in this, and the hierarchy is in this, okay. So, we go one at a time. So, let us look at this. So, here the highest amount of dorsal is in the ventral most area, that is going to activate mesoderm specific genes like twist, snail, FGF8 receptor, not FGF8 ligand, but the receptor, okay. The ligand is going to be produced in the adjacent cells. So, these are being produced here, okay. So, the highest concentration of dorsal is required to activate snail transcription. I am specifically focusing on snail because that is required for us to understand the next layer, slightly dorsal most ventral cells, because they are the ones that are going to make the ectoderm. So, you can imagine the affinity of the enhancers in the snail for dorsal is probably low, and as a result you need very high concentration of dorsal. So, when you move slightly away where you still have significant amount of dorsal, that is not good enough to activate snail, but that activates rhomboid. So, why rhomboid is not produced here? If rhomboid is produced by dorsal, here I have dorsal and dorsal should activate rhomboid, that is because snail inhibits rhomboid production. Okay. So, as a result rhomboid is produced here and this snail is restricted here, and this leads to the production of FGF8, that is going to be the ligand to transduce the signal from FGF8 receptor produced here, and those interactions are going to specify this as glial cells, okay, mesectoderm. Then slightly away you have neurogenic, and then lateral ectoderm, and then you have these ones. Okay, let me tell a little bit about these three. These three are inhibited here by dorsal. If they are not inhibited, they are going to dorsalize the whole embryo. So, while these are activated, they are required for the ventral specifications, and these three are going to activate the dorsal region, and they are inhibited here. And here no dorsal, therefore, these are produced and they are going to specify dorsal ectoderm and the amnoserosa. So, that is an embryonic structure. So, that hierarchy is given here. On the ventral side you have dorsal, that is going to activate these and inhibit these three, which we just saw in that cartoon. This is the same thing here, same information shown to indicate the hierarchy here. And you see the snail is inhibiting rhomboid and dorsal activates it. So, dorsal does not activate snail here because you need high concentration of dorsal to activate snail. So, this is the story of how the dorsoventral patterning is made, and this is not hypothesis, this is real. You see here, these are the expression pattern of different proteins. They are not expressed everywhere. This is expressed in the ectoderm, like this portion, and like this portion is not visible in this. So, therefore, you are not seeing that. So, you see that, and the next one and next one, the muscle cells and then the dorsal cells. So, you see this pattern neatly formed. Okay. So, this is how you start with the symmetrical oocyte and end up with a neatly arranged an embryonic patterning. So, here basically you are patterning the molecular asymmetry, which molecule is going to be where. So, this is how the differential gene expression is playing out during embryonic development. So, this is even more fascinating. If you are already way too excited, this is going to be even more exciting. Okay. So, here we are going to look at a set of genes in a hierarchy. Okay. It starts from maternally produced molecules, okay, maternal effect. These maternal effect means, okay, that is a key word, you need to understand what the maternal effect means. Mutation of this gene will not show the phenotype in that embryo itself. If the mother was mutant, now you will see the phenotype in the embryonic development. So, usually you will see the mutant effect only in the grandchild embryo. Okay, if the mother produced enough of it, let us say you are zygote, both the alleles, okay, the maternal paternal allele both are mutant. So, you are totally having loss of function phenotype, loss of function of that particular gene. Okay. So, YFG is just null allele you have. But if your mother was heterozygous and if she was producing enough of YFG, capital YFG, YFP let us say, and she is depositing lot of that YFP or the YFG mRNA in your zygotic cytoplasm, and if YFP functions only during embryonic development, then that is good enough, then you are going to take shape, you are going to become a normal adult. Only thing is next generation you will not be able to, because your genome is not going to make the YFP. So, as a result you are not going to make functional embryo. Such mutants are called maternal effect mutants. So, mother is YFG over minus. Okay. So, now F1, we are not going to use here, okay, let us use F1, no problem, is going to be 1/4 of them are going to be minus over minus. Let us assume father was a carrier, okay, and so therefore, it is conceivable that you will end up making an embryo like this. Okay, like she will make a gay oocyte that will be negative, right, mutant, and let us say the sperm is also negative, sperm comes from a father who is heterozygous. Now you will end up getting this, but all of this is not happening in isolation, it is all happening in mother. Mother is producing this plenty of YFP or YFG mRNA and depositing in the oocyte cytoplasm. Now let us say this protein is not required for the final neuronal connection in the adult brain, but it is required for anterior posterior polarity in the embryonic stage, and after that it does not play any role. Okay. Any one of the embryonic function you imagine is played by this gene, not later in development. Then the YFP produced from mother's mRNA or YFP deposited by the mother will be enough to take care of that embryonic development. So, this is going to be normal development, okay, normal adult. So, you are going to have YFP protein, let us say YFP protein is still going to be present although the gene is negative, and as a result normal adult. And now this, if you look at its germ cells, they are minus over minus. Now even if it mates with a heterozygous father and produce correct, and let us say this ends up producing minus over plus embryo, this is not going to develop normal because this protein needed to be produced by the mother and deposited there. Assume that the zygotic gene expression does not get turned on in time for it. So, this will end up defective embryo, okay, maybe embryonic lethal. Such mutations we call as a maternal effect. If a mutant phenotype is visible as a maternal effect, meaning I only see 2 generations later, not in the immediate generation, like the homozygous mutant embryo develops normally coming from a heterozygous mother, then you call it as maternal effect. Instead, if the homozygous mutant embryo dies, then that is embryonic lethal, plain embryonic lethal, that maternal word you do not add. So, therefore, you have 2 kinds of genes, one is maternal effect embryonic lethal, another one is called maternal effect sterile, meaning the somatic things will all develop normal, but the gametes will not form right, and they are maternal effect sterile. That is because some of the maternally produced proteins, remember the pole cells form the first. So, they inherit, and their germ cell early development these maternal proteins are required. So, that they are the meanings of those 2 terms, maternal effect sterile and maternal effect lethal. So, okay, so I guess we stop with this definition of what is maternal effect. So, we will continue this in the next class.